I work on efficient clinical trials. I work on trials for rare diseases. So what I want to do over the next half an hour is basically make sure that if you hear the terms adaptive designs and Bayesian designs, you know what these are about and how they can help, potentially help. So if you just think about drug development in general, recent studies have shown that about 80% of phase 2 studies are unsuccessful. Later phase studies, about half of them are unsuccessful. And when you look at likely causes for these failures, it's taking forward treatments that have no hope, studying the wrong patient population, poor precision, optimal dose, maximum tolerated dose, and so on and so forth. So as you can see, even with standard trials, there's already all sorts of problems. Now, when we start talking about rare diseases or relatively rare diseases, there's obviously additional challenges. The limited number of patients is going to mean that either it's simply impossible to recruit the number of patients that are required for your standard randomized controlled study, or if that's not the case, it's going to take ages to recruit the necessary patients. Standard of care might change, and therefore control arms might become uninteresting and unattractive. So what I'll spend some time on is thinking about, can we do better and how can we do better? Now clearly me posing that question implies that I think we can. And two of the potential tools are adaptive and Bayesian designs, sometimes also used in combination. You can also have Bayesian adaptive designs. So let me just explain very briefly what I mean by an adaptive design and give you a short illustration to show how potentially this can help. So the idea is quite simple. We try to modify an ongoing trial, which, by the way, when I did my master's degree, I was told to never do that. But there you go, 20, 30 years on. We've now found methods that allow us to do that. We do that by design, so we think a priori what sort of modifications might be of interest, and I'll give a couple of examples in a little bit, in order to get a bit of flexibility in our design, in our decision making. And the key part is, of course, we want to do that without undermining the study's integrity so that the results from this study can be interpreted in the same way as your standard clinical trial, your standard randomized controlled study would be. So, current standard fixed sample design, randomized controlled trial. Basically, the team doing the investigation starts planning the study. The statistician at one point comes into play and is going to say, well, we need about 150 patients. So fix n the sample size. Then you start the study and you wait, you recruit, you wait until you've observed all 150 patients, at which point you then look at all your data and make a decision. An adaptive design, in contrast, starts still off with planning, fixing a design. Notice I'm not talking about fixing a sample size here anymore. You start the study, but rather than wait possibly for years until you have recruited enough patients, you look at the data a couple of times at so-called interim design points. And at each of these design points, you can make adaptations to your study, preferably pre-planned adaptations. So for example, you could stop the study, either because the treatment works so fantastically well, we don't need the remaining 100 patients. The first 50 were enough. Or probably more likely, the treatment just doesn't work. So why waste any more resources, any more time on evaluating something that after 50 patients we already know is not going to do anything? We can change the sample size. For example, a lot of these calculations rely on assumptions. These assumptions may obviously be incorrect because you're thinking about a different population and so on and so forth. And so these designs give you the opportunity to correct for these incorrect guesses. You can change the allocation ratio. What do I mean by that? How likely is it that a patient is going to be recruited, is going to be assigned to treatment A versus treatment B? And ideally, you would set it up in a way that the better the treatment works, the more likely it becomes that the patient gets allocated to that particular patient, rather than having 50% of your patients on the control group. If you already think you have compelling evidence that the treatment seems to be at least not worse than the control group. Or you can do a bit more extreme adaptations, dropping, adding arms, doses, and so on and so forth. So here's a very simple example of how an adaptive design could look like. So here I'm thinking about a study where I have 3 different treatments that I might be interested in, and I'm going to use a control group. So each of these T's corresponds to my measure of effect of treatment 1, 2, 3 against my comparator, against my standard of care. And then after a certain number of patients, I look at my data, and in this case, I compute my measure of effect. How likely is it that this treatment is that either of these treatments is doing us any good? And in this case, because there's insufficient evidence for the second treatment, it falls in the red shaded area, we give up on that treatment. It doesn't look like it's promising enough, it doesn't look like it's helpful enough, and therefore it's out. No more patients randomized to that particular treatment. For the other 2, we are in the continuation region. We are in the part where our conclusion is, well, it looks okay, but we don't know quite yet whether we have enough evidence. So we continue on. We recruit another batch of patients to those 2 treatments only, 1, 3, and control group, standard of care group. And if you want, as I said, you can alter the allocation ratio to these different arms as well. Then you look at your data, you find at the second interim analysis, the second look at the data, still insufficient evidence to stop the study, insufficient evidence to say one treatment is better than our standard of care. And in our third look, we find a winner. We get one of our effect estimates that says we're better than control, and therefore we can stop and celebrate. Now, the advantage of doing that is, first of all, we started off with 3 different treatments against the standard of care. So, in terms of randomization, you have a much larger chance of getting one of the new agents in this study. In addition, we gain efficiency because rather than having 3 studies, each of them having a control group, with 1 study with 1 control group. So we've reduced the number of patients in total that would be on the control group. We saved by dropping arms that weren't promising, and you can see there is still a gap, which suggests that the trial could have gone on, but we didn't have to. We had the conclusion already before, so we never needed the total sample size, the maximum sample size that was required for that particular study. Okay, so that's just a very, very, very brief overview of how an adaptive design could work. Next, I'll just briefly introduce mostly through a little illustration what a Bayesian design is. And in the recent past, we've used quite a lot of these Bayesian designs in the context of small populations, rare diseases, in order to augment information with knowledge that's already there. So how does a Bayesian design work? It's pretty simple, really. We start off with what we know already. So that might be other studies, that might be expert opinion, anything that you believe is relevant for your study. Then you collect your data for your study, and then you combine these 2 pieces of information, the prior information, the thing you knew before, with the evidence that you've collected. So you could think about the prior opinion capturing the control group, and the new data being the data on the experimental treatment. You combine them in order to make a decision about the treatment. So I just want to very briefly illustrate how such a trial would work. So you are now my experts. So I'm not going to pretend I have historical data, other data sources that I want to use. You're going to be my experts that are going to tell me about what you think the chances that my new treatment works. Okay? So I'm pretending I'm planning a study for a new treatment. All of you have some experience with that treatment or other relevant treatments. And in order to emulate, in order to illustrate your knowledge, I got a little glass vial in here that has pink and black blocks. I'm going to flash that at you for about a fraction of a second. And your task is to estimate the proportion of pink blocks. And so what we're going to pretend is that this is your knowledge, your belief that this treatment will cure, will help. Okay? Everybody ready? As I said, it will only be a fraction of a second, otherwise it would be too easy. Ready? Right. So now, I know, but we want to make it interesting, right? If I show it too long, you'll get the right answer straight away, which isn't going to make an interesting example, I'm afraid. So, the first thing that I, as the statistician, are going to have to do is I'm going to have to extract your knowledge, try to find out what you believe the proportion of patients that will benefit from my new treatment is going to be. And I'm going to put that in a mathematical model without showing you the model. So, I got two questions for you, and I'm sorry if that's hard to see. I'll start off with the first one. Okay, so the first one is, what is the most likely probability that a patient gets cured? Translated to our example, what's the proportion of pink blocks in my little glass? No wrong answers here, just give me a couple of guesses. 50%? 70%? 0%. Okay, right. So we got a broad range. And so if we were to do that in reality, we'd have about 10 experts in the field sitting in front of you, asking them the same question, and we get about 12 different answers. At this stage, our task is come up with a consensus opinion what that proportion is going to be, and there are some form processes that we can use in order to come up with an estimate. So based on what I've heard, I think something around 55% sounds reasonable. We had 50, 70, 80, and 0. So if I just take an average, I'm assuming that's somewhere close to 55%. So that's my first piece of information that I need from my experts. The second piece of information that I'll need is, what's the proportion that you're pretty sure the response rate is below? So what I'm looking for is an upper limit. You're pretty sure it won't be any larger, the success probability, than that. And by pretty sure, I mean 75% sure. Any guesses? 50%? So the first thing to notice is I'm looking for an upper value, so it's got to be larger than what we've guessed before. Because before we said, well, we're pretty sure that on average, 55% will respond. Now we're looking for an upper limit that we are pretty sure we won't exceed. 85. So 85, 70, 75. So again, we would go through the same process with our experts and try to come up with a consensus opinion. Okay, so I'm going to change back to my little graph here. So if we say 80% for the upper bound and 55, hoping that my program actually works. Always the same. If you illustrate something, you can be sure that it won't work. Just give me a second. Right. Let's try that again and see whether the program... No? Right. Okay. So this is my guess. So this is my guess for what the distribution a priori, before I collect any data, could look like. So here, that basically, how to read that graph is, that's the probability of success between 0 and 1. And that is how likely that particular outcome is. And you can see, based on the answers that I've inputted, we don't seem to know a whole lot about the effect of that treatment at that point, right? Because it's a very, very wide distribution. But obviously, if I had shown you the vial a little bit longer, maybe the distribution would have looked something like this. So you would be pretty sure, 55% sure, that the response is 55%, sorry, very sure. We're pretty sure that values below 25% are not likely. We're pretty sure values above, say, 80%, 85% aren't very likely. So this is my prior information. This is the information that I had before. And as I said, that could be expert opinion, that could be other data, that could be other studies, anything really that you think is relevant for the study you're planning. And so the next step is then to collect some data. So what I'm going to do is just I'm going to randomly draw, or let somebody randomly draw patients. Say 3 patients. Can I get somebody to just pick 3 patients? And remember, pink corresponds to that patient responding, having a positive effect. So we got 1 responder, 1 pink, and 2 and 2 non-responders. And you can see that now that I've combined the data with my prior information, my distribution, now in green, has changed. You can see it's moved downwards because I've only observed a success rate of 1/3. I thought it was going to be 55 initially. That's why I see the move down. But if you look very carefully, you'll see that the distribution has also gotten narrower. So we've learned something. We're now pretty sure that values above 75% are very unlikely on that treatment. So how would you continue with that study? Well, you would just collect data, collect more and more and more data, until you're happy that you have enough information to have a valid conclusion. So, if I show you that green distribution, the first question would be, do we have enough evidence to conclude that our new treatment works well? Well, I guess the answer is depends, right? I mean, if the alternative has 0% success rate, yeah, I'll be convinced. Reality, of course, fortunately, that's not the case, because our comparator will be somewhere in that wide area. We probably need more data. And so we would continue the process, collect more data. And if I pretend I've heard and observed 30 patients, and let's assume that just under half of them have had a positive response, you can see again the distribution has shifted because I had less than a half. We started off with just over a half in our initial guess. But the distribution got narrower and narrower, and again, I continue until I'm satisfied that the amount of information I have is conclusive, is sufficient for me to say, yes, my treatment is better than whatever I'm comparing against. So that's a Bayesian design in a nutshell. And as I said, there are different ways that you could incorporate that information. We've heard just before that there is evidence from other cancers that immunotherapies work very well. Maybe that's your prior information that you want to use. It might be that you have a lot of information about the standard of care arm, and therefore you could use that information to inform your prior distribution, and so on and so forth.
Can I ask a question?
Of course.
collecting data like you are explaining now means that you are getting more and more patients involved in your trial. Would that mean that you are steering the outcome? You're looking for as much patients that you get the satisfaction, an outcome that gives you the satisfaction. It might be biased.
Yes, so it's a very good comment. So I mean, it is certainly the case that whatever the information is that you've put in, it will be reflected in your final distribution. The fact that you continue doesn't matter all too much, because if the truth is 20%, collecting more patients isn't going to help change the fact that only 20% of patients will respond. So bias in that sense is less of a problem. But obviously, if your prior information was incorrect, then that would shift slightly. You can actually work out how much information the prior distribution contains. So for example, in my little example, the prior information was worth just under 10 patients. So it was as if we would have seen 10 patients in our study. Yeah, did that answer your question? Yeah, so in a Bayesian trial, you determine your prior, and as I said, that's probably the trickiest part for the statistician. Then you update your prior with your data, you augment your prior with the data that you've collected, and you ask yourself the question of, when do I have enough information to make a solid conclusion based on prior information and collected information? So, just to briefly wrap up, really, I'm not saying that randomized controlled trials don't have a role to play. They are the gold standard. There are plenty of disease areas where this is the way to go, where there's no problem with recruitment, there is no problem with the standard of care arms having good response rates and so on and so forth. But especially for rare diseases, Bayesian and adaptive methods have a big role to play. They're helpful in reducing, in gaining efficiency in conducting the study, which for you means treatments coming to the market quicker, hopefully. And Bayesian method in particular can be helpful to augment data, augment information with data that you have already a lot of confidence in. Thank you.
Any questions?
I guess it's really a question of, the theory may be fine, but does the FDA and the European equivalent buy any of this?
Yes.
And because that's the key thing, is will they accept a changed trial design as much as the theory of it?
Yes. So both EMA and FDA have guidelines out on adaptive designs, saying that there are adaptive designs that they're happy to use, that are methods they're happy to support. There are some adaptations they will not accept. For example, they will not accept changing your endpoint. So what are you actually looking at on the basis of the data that you've collected? But the sorts of things that I've illustrated, they are happy with. The sort of little toy example that I've shown, we've used in a couple of rare rheumatoid arthritis type diseases, and again, both EMA and FDA have been happy with them. As I said, the randomized controlled trials are the standard, so you can't just go in and say, I'm going to use one of these methods because I feel like it. You need to justify why this is necessary in your specific study. But in general, yes, they do buy, and they do accept these things.
But typically in this country, NICE will want to randomized control before it will approve?
Again, not necessarily, not strictly. It depends on the case you're making.
And can you incorporate real-world data from registries into your Bayesian design?
Yes. Where your prior comes from doesn't really matter. as long as you believe it's relevant for your question. Now, there is a risk. So, of course, if in my little toy example here, I know that the truth is just under 50%. If I would have started off with a prior distribution that maybe looks like this, I'm going to negatively influence the outcome of the study. So you need to be sure that the data that you want to include are going to be relevant. But where the data sources or where the information comes from doesn't matter as long as it's relevant. And that's probably the biggest argument you'll have with regulators, justifying why this is the right source of information to use. So I've talked about these rheumatoid rare conditions. For example, there we had to be very precise about defining who we called an expert. We had to be very precise that we captured experts from all around the world, not just from the UK, for example. But as long as you satisfy these things, the regulatory agencies can buy into these things, yes.
I've got a question myself, Thomas. Obviously, this is going to be potentially very valuable for us because we've known things have taken a very long time. We've got people dying, you know. So what can we do as a patient group to influence companies and pharmaceutical companies and say, no, we really want you to do an adaptive design because it will mean getting to market quicker and getting people cured quicker? Because we suspect we've got drugs coming up in the pipeline from various research projects. We want to anticipate this. So where do we go? Do we speak to you or what do we do?
So large companies will have expertise in these sorts of methods, so they will not be foreign to them. What you're going to possibly see is a hesitance of the company to use these methods because it's just not what they've usually done. And so what you need to do is use your combined voices to say, well, but this is what we want and this is why we think it is helpful because it will help recruit patients, it will help to answer the questions at hand much quicker. So opinion publications, for example, are helpful in that respect, for example. I think there was one.
In theory, would a Bayesian design be more useful for something like DELCAT or for something like IMCGP?
Sorry, I'm not a subject expert, so I'm not sure I can answer that question.
DELCAT is something that has already been tried in patients, and you can already see results. Whereas the other one is something that is still a new drug.
Mm-hmm. Yeah, so the more information you have, the more efficiency you're going to gain. So if there is already some experience with a treatment, you'll gain more with a Bayesian design than if you don't know a whole lot at the beginning.
So it's mostly useful for speeding up approval for a drug that has already been tested in a conventional trial?
Not necessarily, because you can, for example, as I said, the standard of care, you will have a lot of information about that already, no matter how new your new treatment is. So you can use the historical information on the standard of care in your prior distribution. And it's quite possible to say, I have a lot of information about one arm in my study, and I don't know a whole lot about the other arm, and you still gain by using just the control arm information.
So in the previous presentation, we had an example of a trial where people were allowed to switch, and that made the trial less statistically significant. So my question is, you're talking about this adaptive design. In principle, can you design the trial to have this interim design points? Because speed is of the essence and we want to get results faster. Can you have these interim design points designed so that after 3 months you allow the switch, but it does not become less statistically significant, and it's still something that will show the difference that we want to investigate?
The short answer is depends, as you probably feared. it depends on whether the first set of treatment changed the disease progression or the status of the patients. So if you can convincingly say patients that switch to the new treatment at an interim analysis are comparable to patients that would have gotten the new treatment from the start, as long as you can do that, that's okay. How to show that is a bit more tricky, and this is certainly something where regulators are very picky. Not, again, I mean, they have accepted that in other contexts, but it's certainly not something that they would routinely accept.
There is a definition of rare diseases. Can you remember what it is?
There are some advantages to being an orphan disease in terms of regulation. I can't remember what they are.
Yeah, I must admit I can't remember what the incidence rate needs to be for it to be classified as a rare disease. I want to say 1 in 40,000 possibly.
100,000 new cases in America per year.
Yeah, I must admit I can't remember. The orphan disease designation does help. In the sense that regulators tend to be a bit more flexible in what they accept in these in orphan diseases.
Any other questions? Okay, well, thank you ever so much. I think you will actually make the train home.
Great.